Okay. Thanks, Will, and the organizers. I think Maria has a good start. I'm gonna, as Will has introduced, I'm gonna give a modeling talk today, and I'll share a story how to use the mathematical models to extract the insights from cross-reading experiments. So, cross-feeding is a phenomenon that some microbial species feed on the molecules produced by other microbial species. The question that we are interested in is, given the population level data, such as the population dynamics, the population density and the metabolic concentration, can we quantify and measure the metabolic exchange fluxes from the data? So why cross-feeding? First, the cross-feeding is prevalent in nature. So if we link the species by the metabolites that they can transport, as I shown in the figure, that our human gut microbiota can become, super dense metabolic network. So it's very prevalent that the cross-feeding can occur in nature. Another reason that we started cross-feeding is that it's a great challenge in microbial ecology to infer the metabolic fluxes from data. The reason is that what we can actually measure from the data, such as the metabolic concentration change from, over time, is only the net flux that resulted from contributions of multiple species. So for any metabolite, such as, the star I showed here, it can be secreted and uptaken by many species at different rates at the same time. So it's, cross-feeding is high order interaction, and it's hard to disentangle the, individual contributions from the species. So here we hypothesized that to infer the metabolic fluxes for the individual contributions, we have to combine the mathematical models with the experiment data. The model that we use is similar to resource transfer model, but the major difference is that we allow a simplified and coarse-grained metabolic network within each cell type. So we allow two types, we model two types of molecules, the substrate as shown by the S, S1 and S2, and the metabolic building blocks, the M1 and M2. And we also consider three types of reactions. The red arrows represent the resource utilization, the uptake reaction, which is modeled by the monod type kinetics. The blue arrows represent the resource conversion, and we modeled the resource conversion rate as as a way similar to the flux balance analysis. So basically, we assume that the intercellular molecules have a really fast dynamics, and if they reach steady state, the equilibrium very fast. And the consequence is that for each intercellular metabolite, the total influx would be equal to the total outflux. And we have parameters for those unknown fluxes, and those parameters should be quantified from the data, fed from the data. And also we consider the biomass flux reaction, and we use the mean function, and the underlying logic is that the growth rate should be limited by the most limiting nutrients among all of the metabolites and the substrates. And we also consider the catabolite repression and the growth inhibition. I will give two examples to show the utility of the model and how to use how to combine the model and the experiment to quantify the exchange reactions, metabolic fluxes, and how to use them, parameterize the model to predict new experiments. The first example is a simple engineered community of two amino acid auxotroph of coli. So in the red strand, that the single gene has been deleted in the leucine biosynthesis pathway. So it's a leucine auxotroph, and the red cell type is has the single gene deletion in the lysine auxotroph in the lysine biosynthesis pathway. So both the red and the blue cells, the leucine and the lysine auxotroph, cannot grow in monoculture, but they can potentially grow in co-culture by using the amino acid that their partner can produce. Our model is, was developed to mimic the experiment using the framework I just described in the previous slide. And we showed that by fitting the data, we can find a set of the biological relevant parameters to reproduce the data very well for both the monoculture and the co-culture data. So given the model, what type of experiment can we produce? I mean, what type of, the new predictions can we make? So I will use one slide to show that. I put the model in a chemostat with dilution rate of equal to 0.1. And I asked the question, by supplementing amino acids to the system, how does the ecological social interactions change by adding amino acids? So naively, we can expect that the two auxotrophic strands depend on each other to grow. So their relationship should be mutualistic. But our simulations show that by adding amino acids, the relationship, the mutualistic relationship can be destabilized and change to other ecological interaction types. For example, if we add a lot of leucine, the mutualism would become the competitive exclusion. Or if we add a lot of lysine, the mutualism could become parasitism. And we determine that the differential ecological outcomes is ultimately dictated by whether the lysine or the leucine auxotroph are limited by the glucose or the amino acids they cannot produce. For example, in the R2 region, both of the lysine and the leucine auxotroph are limited by glucose, so they compete for the same nutrient, and the competitive exclusion would occur. And we can make a further prediction by adding the lysine and the leucine together. So this is equivalent to draw a straight line in the diagonal direction. And our simulation predicts that the ecological interactions change in the order from collapse to mutualism to parasitism to competitive exclusion. And this order has been validated by an independent experiment from Jeff Goff's lab from MIT. And but in their experiment, they engineered the yeast strains and used different amino acid auxotroph. But overall, I think the validation from the experiment suggests that our model have captured the correct ecology that should be generalizable across different biological systems. The second example is the 14 amino acid auxotroph of E. coli. So it's similar to the lysine leucine auxotroph, but we have 14 strands that have each of the amino acid auxotroph has one gene deletion in one amino acid biogenesis pathway. And I listed the gene that has been deleted here. And the model is also similar, but the model can be extended by incorporating the fact that each of the amino acid auxotroph can secrete 13 amino acids that can be uptaken by other amino acid auxotrophs. We use two pieces of data to fit the model parameters. The first piece of data is the growth fold, the final density divided by initial density in the pairwise spatial culture. We show that the model reproduces the data pretty well when the observed growth fold is larger than 1. The discrepancy here is because we don't allow the bacteria cells to die. I mean, the population density will never decay in simulations, but it's possible in the experiment that population density decreases because in this range that the OD, the initial OD is very low, and it's the OD measurement is highly uncertain, and it's possible that there's some fluctuations and population density decreases over time. The second piece of data we use to fit the parameters is the daily serial dilution data, the time series data from 5 communities. One is the community of all of the 14 amino acid auxotrophs, and 4 different 13 auxotroph community with 1 amino acid auxotroph left out. And the dots or the circles of the data, and the lines of the simulations, and except for the no methionine community, I think the model predicts the, fitted the data with a decent accuracy. So with the model, we can report the quantitative values of the amino acid secretion. So each value in the matrix represents how much carbon salt has been lost in secreting the amino acids in the y-axis by the amino acid auxotroph in the x-axis. So for example, this value, the 13.32, represents 13% of the carbon salt has been lost in secreting the threonine by the methionine auxotroph. So we see a great diversity across the 14 amino acid auxotrophs. And some amino acid auxotrophs are more cooperative, such as the methionine auxotroph. They secrete about 36% of carbon in form of different amino acids. But other, the tryptophan auxotroph is only secrete 1%, so it's less cooperative. We can use the model to predict the steady state composition. So here I showed the progression of the simulation from, and it's a 2 replicates of experiment together. So the simulation shows that maybe after 4 or 5 days, that the simulated composition of the microbial community converged to a steady state composed of 4 strands: the isoleucine auxotroph, lysine auxotroph, methionine auxotroph, and the sterolene auxotroph. And I think the experiment validates the simulation in at least 2 aspects. So first, there exists a steady state. Second, the steady state composition is composed of these 4 amino acid auxotrophs. So the take-home message is that we use the coarse-grained metabolic models to infer the metabolic exchange fluxes from the data, and it's sufficient. We don't need more complex models such as whole genome reconstruction models. And also, we showed the utility of the model used 2 engineered communities, so that the parameterized models could facilitate the design of the synthetic ecosystems towards more predictive population dynamics. And there's definitely some caveats. For example, the parameter fitting for the 14 amino acid auxotroph, that we found that we cannot fit the parameters using the optimization algorithm alone. So we have to use both the optimization algorithm with some manual fitting to guarantee that the parameters both fit the data well and are biologically relevant. Also, the 2 examples I shown in this talk, amino acid auxotroph, we know that they cross-fit by amino acids, but for a given microbial community, a random community, that we have no idea what kinds of cross-fitting interactions could drive the population dynamics. So one solution could be using the genome scale models, the whole genome reconstruction models, to predict the possible cross-fitting interactions, so that we can determine the topology of the interaction and develop a model based on that topology and fit the parameters from data. So I'm gonna thank my lab member, not my lab, my colleagues in Viva lab member and our collaborators. So I'll be happy to take questions, and thanks for attendance of my talk. Yeah, thanks, Will. Yeah.
All right. Yeah. Thanks for a great talk. Mike Blazenin has an interesting question about sort of model choice, and he was wondering if you could discuss the motivation for using this model formulation rather than a simpler one like the consumer resource model. You touched on that a little bit at the end, I think, about sort of the intermediate between genome scale and simpler ones, but can you expound on that a little?
Yeah, for the simpler models, we tried Lactobacillus, and we showed that I didn't show in the slides, but for the 14 amino acid auxotroph community, we tried Lactobacillus, and we showed that the Lactobacillus cannot capture the data, even if we use the microbial chain monocolor simulation to optimize the parameters. For the resource consumer model, we don't use that as a reference, but I think the difference is, as I described in the talk, that we allow the metabolic details, although in the coarse-grained manner, but we allow the metabolic details that can capture the biology and the metabolism of what's happening in the cells. But the resource consumer models basically, combine the resource conversion and the transport in one reaction. So it lacks the flexibility to incorporate the metabolic details.
That makes sense.